and next we have Dr. Christos Papadelis from Cook Children's Medical Center, who's going to be telling us some about his work on electrophysiological biomarkers of GABA in Dravet syndrome.
Okay, good evening, everybody. First of all, I would like to thank for the invitation. It's a great honor to be here. I know it's quite late, and I know that my talk is a little bit like more into understanding the underlying pathophysiology in children with Dravet and what's going on in the brain. I will describe like all the mechanisms and the tools that we're trying to develop for assessing the levels of the GABA in children with Dravet. This is a study funded by Encoded Therapeutics, and these are my financial disclosures. So, as we know, like we are dealing with a genetic form of pediatric epilepsy, affects approximately 1 to 15,000 live births, and we know that like 70 to 80% of patients with Dravet, we have recognized these mutations, the SCN1A gene, that encodes like the AE1 subunit of the voltage-gated sodium 1.1 channel. So let's go like to understand a little bit better what is this channel and why this is important. The voltage-gated sodium channel like play a critical role in the initiation of what we call axial potentials in the neurons and in the regulation of their excitability. The sodium channels, they are located at the axon heel locks and across the length of the neural axons. And during the resting period, like the channels are initially closed, so there's no any kind of influx or outflux across the neuron's membrane. So when we have a nerve pulse that arrives, like the sodium channels, they are activated and allowing like positively charged sodium ions to enter the cell. And following the activation, the channel does not open in response to a new signal for a brief period of time. And this critical sequence of events like dictates the propagation of axial potentials towards one specific direction. So studies on patients with Dravet, they have shown that mutations in the SCN1A gene typically produce non-functional channels, sodium 1.1 channels. And then these non-functional channels like will cause reduced sodium current, and that's less sodium depolarization and inexcitability in the neurons. So the main idea, and that's the main hypothesis like behind the Dravet syndrome, that is the result mostly of what we call de novo SCN1A haploinsufficiency. Actually, it's a genetic term that indicates a situation where the total level of the product, the gene product produced by the cell is about half of the normal level, and that's not sufficient to permit the cell to function properly. However, the problem we have here is like there's a kind of a paradox, like it's a surprise that reduced expression of a gene that encodes an essential depolarization current like cause epilepsy. And this because like reduced sodium current like should lead to inexcitability rather than hyperexcitability. So in order to explain this kind of paradox, we have formulated alternative hypotheses. Most of these hypotheses are based on recent animal studies. One of these hypotheses, like what we call interneuron hypothesis, it makes an assumption that the seizures in Dravet are the result of what's called selective defects on the GABA interneurons, which produce a network that lacks sufficient inhibitory tone. We can think as kind of a car that works like properly, but the brakes are broken, so there's no any kind of inhibition in this kind of activity. So let's talk a little bit more about the GABAergic system. The GABAergic system is one of the two important amino acid systems in the human brain. The glutamate system is responsible for fast signaling excitation. The GABA system is responsible for fast signaling inhibition. So it's like to have a big, large off switch in an electrical circuit that responds like very quickly and has a global effect, since nearly half of all synapses in the brain, they express some kind of GABA receptor. So let's see how the GABA neurotransmitters, they act. They act at the synaptic level by modulating ion channel activity to two major postsynaptic receptors, the GABA-A inotropic receptor and the GABA-B metamotropic receptors. So upon GABA transmitter binding, the GABA-A receptors, they open, allow negatively charged chloride ions to flow into the neuron, make it more negative or more hyperpolarized, and that's less likely to generate any axial potentials. Upon GABA transmission binding, the GABA B receptor is also activated, opens, and allows like positively charged potassium ions to flow out from the cell, making the postsynaptic neurons hyperpolarized, and that's less likely to generate any actual potentials. Oops. Sorry for that. Okay. So the proper function of the central nervous system depends on the balance between the excitatory and inhibitory neurotransmitter systems. The excitatory system is regulated by glutamate, as we said before. The inhibitory system is regulated by GABA through the interneurons. And these interneurons, they control the flow of information and the synchronization of the cerebral cortex, and can be seen in several brain regions such as the amygdala, the hippocampus, the hypothalamus, and the prefrontal cortex. So the problem we face now is that the GABA cannot penetrate the blood-brain barrier, and that's its measures through the blood tests are not reliable, so they do not reflect exactly the levels of the GABA in the brain. So there's a recent interest now to develop non-invasive biomarkers, which are based on brain neuroimaging tools, which they can provide like an indirect measures of the levels of the GABA in the brain. There are several neuroimaging methods which can help us assess in an indirect way the levels of the GABA in the brain. And among them, today we'll talk about like what's called magnetoencephalography or MEG, the high-density EEG or high-density electroencephalography, and the magnetic resonance spectroscopy or MRS. The MEG or magnetoencephalography is a non-invasive neuroimaging technique. It measures the magnetic activity generated by the human brain. It actually presents several advantages compared to other neuroimaging methods, particularly for children. The child sits comfortably in an armchair, places the head inside of this helmet. Inside of this helmet, we have several sensors. They're called gradiometers or magnetometers. And we measure the activity of the brain through magnetic fields. Actually, what we have the MEG measurements. They are based on the phenomenon of electromagnetic induction, according to which like any kind of changing magnetic field generates in a nearby coil an electrical current. So we have like thousands of neurons in a focal area of the brain. These neurons, they all simultaneously active. They generate an electrical current. This electrical current generates a magnetic field, and this is what we measure outside of the patient's scalp with the MEG. A technique that is comparable to the MEG is what's called high-density EEG. This measures the electrical activity of the human brain. Modern high-density EEG systems, they allow the recording of up to 256 electrodes covering the entire child's head without even the need to apply for any kind of gel for people who have experienced EEG recordings. This kind of EEG nets, they accommodate like sponges. They're attached to each of these electrodes. We soak the net inside of a saline solution in salty water, actually, and then we place the net on the child's head and we start performing the recordings. This is like the quite unique setup we have at Cook Children's Medical Center in Fort Worth, allows the simultaneous recording of magnetic and electrical activity of the child's brain with more than 550 sensors that cover the entire child's head. So there are three basic neurophysiological mechanisms which are candidates to generate an electric and magnetic field that is measurable outside of the scalp. We have axial potentials, which are traveling away from the soma. We have postsynaptic currents at the apical dendrites of the neurons, and we also have backpropagated spikes into the dendrites. And among these three mechanisms, the postsynaptic currents, they are the one that mostly contribute to the EEG and the MEG signal. Although recent evidence, they have shown that also axial potentials, they may contribute to what's called high-frequency activities or high-frequency oscillations. Magnetic resonance spectroscopy, or MRS, it's another neuroimaging technique. It allows to examine the human tissue for the presence and concentration of different metabolites, like the GABA. The advantage is it's conducted on the same machine as the conventional MRI. So if you bring the child for a normal MRI, they can have also a magnetic resonance spectroscopy scan. The problem with our studies, because we do research, we cannot sedate the kids, so it's really difficult for us to record this kind of data in kids with Dravet without sedation. A first step towards tools that allows to measure the levels of the brain GABA non-invasively. It is the observation that drugs such as the benzodiazepines or benzos, which potentiate the GABA-induced chloride influx, affect the oscillatory pattern of the EEG signal. So although benzos, they are sedated drugs, there are recent animal studies showing that an increase in what's called beta and gamma activity is higher activities in the EEG signal, and this is possibly through the action on the GABA-A receptors. So the question is now how we can induce this kind of frequency-specific changes in the human brain and how we can measure them with non-invasive tools such as the EEG or the MEG. Early studies on patients with implanted intracranial EEG electrodes, they have shown task-specific high-frequency activity between 40 and 150 hertz in the raw signals in response to visual stimuli, which were presented to the patient. Later, by using more advanced neuroimaging methods, such as the MEG, other studies, they have shown gamma band activity in frequencies approximately 60 hertz induced by the presentation of visual stimuli in healthy adult subjects. And later, another MEG study from another group of researchers, they also showed evidence of gamma-induced activity in frequencies between 60 and 95 hertz in the primary somatosensory cortex of healthy adults in response to noxious stimuli. In order to measure this activity non-invasively, we need to adopt modern neuroscience experimental designs. Here we see, for example, the visual experimental setup we have in the MEG lab at Cook Children's Medical Center. We actually present to the child several visual stimuli in a screen that is located in front of the child, and we measure the brain activity in response to this kind of stimuli. For the enhancement of the gamma-induced activity, we present flashing checkboards that is well known that generates the strongest possible cortical response within the visual cortex. I will try not to go into details, but I would like just to highlight this kind of unique technology that we have in order to assess this kind of weak brain responses, which can be easily recorded with invasive implanted electrodes. We need to increase what's called signal-to-noise ratio, the quality of the EEG or the MEG signals that we record non-invasively. And to do so, we adopt a methodology that is based on electric and magnetic source imaging. It involves the solution of complex mathematical problems. We actually initially co-register the location of the different EEG and MEG sensors with the patient's head scalp, which was extracted from the MRI. And based on this information, we develop a head model that approximates the electromagnetic properties of the child's head. So by using this kind of model, we're able to estimate non-invasively what we call virtual channels. So this is high-quality EEG and MEG signals from the brain within one specific region without having any kind of invasive procedure. By using these two methodological approaches, my team has shown in recent studies that we can detect non-invasively what's called high-frequency oscillations. It's activity between 80 and 250 hertz, called ripples. This kind of activity can often be seen overlaid on interictal spikes and seen mostly in patients with refractory epilepsy. This is a study from a MEG group in the United Kingdom in healthy adults. They showed for the first time evidence that the amplitude and the frequency of the gamma-induced responses to visually presented stimuli, which were recorded with a MEG, was positively correlated with the resting state levels of the GABA in the brain that was assessed with a magnetic resonance spectroscopy. And actually, they performed a similar experiment with function MRI, and they found that the bold amplitude of the hemodynamic response in the primary visual cortex was negatively correlated with the levels of the GABA and the gamma frequency. That's a study from another group from CHOP in Philadelphia here in the United States. They showed similar findings in the primary motor cortex with the MEG. By using this approach I described before with the virtual sensors, they found what's called movement-related gamma synchrony and the frequencies of 80 hertz. That was in response to finger tapping. They asked the participants to move their finger, and then they record the brain activity at the same time. More recently, a group from Germany, they examined the correlation of the levels of the GABA with the gamma-induced responses in the auditory system. By using the technique we described before, magnetic resonance spectroscopy, they estimate the levels of the GABA in healthy adults and on a large voxel covering the primary auditory cortex. And they used an EEG later in a passive listening task to record brain responses to auditory stimuli from healthy adults. In this study, they didn't manage to successfully correlate this kind of frequency and amplitude of gamma activity within the levels of the GABA in the auditory cortex, but they replicate the previous findings that they were induced some kind of successfully gamma activity in the auditory cortex. This is the only study exists on Dravet syndrome so far, and this is what we're trying to enhance here. It's a study from Europe. They tested the hypothesis that brain oscillator activity in the gamma frequency band is altered as a result of GABA dysregulation in the brain of patients with Dravet syndrome. Actually, they examined like 13 children with Dravet with EEG. They use an auditory experiment. They provide beeps and the children, and then they compare the EEG findings with them from a group of a non-Dravet epilepsy and a group of healthy controls. So as you can see here, like they see in these time frequency analysis plots, I will explain later what this means, like that in the kids with Dravet, this kind of activity was absent compared to the other groups, and this indicates a dysregulation in the GABA in their brain. To summarize also these findings, this European group, they observed like weaker or no responses in the frequencies between 1 and 120 hertz for patients with Dravet. That was compared to both healthy controls and children with non-Dravet epilepsy. And these kind of findings, they indicate a suppression of gamma activity in children with Dravet compared to the healthy controls and support our hypothesis that the GABA suppression plays a significant role in children with Dravet syndrome. This is a paper my team published recently. It presents findings from another disorder, not on Dravet, but it's another GABAergic disorder. It's called SSA-DHD. It's much more rare than Dravet. It's an ultra-rare genetic epilepsy. Actually, there are only 300 families in the planet. And we fly some of these kids to Boston Children's Hospital. That was my previous institution where the study was performed. This disorder like has some similarities with Dravet. This is what we performed there. We hypothesized that these patients with SSA-DHD, they will also have like altered somatosensory responses in the same frequencies in the beta and the gamma frequency bands compared to the healthy controls. In order to test our hypothesis, we recorded what's called somatosensory evoked potentials. We use high-density EEG, as you saw before. That was a previous stage of the technology. We used like EEG with gel. Preparation was like tedious, but it was quite successful. In order to elicit this kind of somatosensory responses, we provide to the fingers of the children pneumatic air puffs through these plastic tubes. They were attached to their fingers. And then we initially, based on the patient's MRI, we estimate again what I described before, these virtual channels which were located now in the primary somatosensory cortex, because that was our interest in this experiment. We also assess the levels of the GABA in the blood for these patients, and we correlate the findings between the blood findings and the findings from the brain. So here you see what is called time frequency analysis plot. I will try to explain that. It's here on this is like the group of typically developing kids, and this is a group with SSA-DHD. When you see the zero line here is when we provide the air puffs to the kids. And what you see here with red color is when the brain responds to this kind of stimuli. On the x-axis, you see the timing from 0 to 500 milliseconds. And then on the y-axis, you see the different frequencies in the brain where this activity occurs. So what we observed that kids with SSA-DHD, they have a suppressed response to this kind of stimuli, mostly in the gamma and the beta frequency band. We observe also a very strong negative correlation between event-related power synchronization, what I explained before, in the beta band and the levels of the GABA in the blood. So, and from this graph, you can see very clearly a classification between the patients with SSC-DHD and the healthy controls in terms of like beta synchronization and the levels of the GABA in the blood specimens. We observe similar findings for the gamma activity, a moderate correlation between this event-related power synchronization in the gamma frequency band and the levels of the GABA in the blood among participants. So based on all this evidence that I presented so far in my previous lab, we initiate a project at Cook Children's Medical Center together with Dr. Scott Perry. This project is funded by the Encoded Therapeutics. The initial idea was like to develop a non-invasive tool that allows the assessment of the levels of the GABA in the brain and correlate these kind of findings with the levels of the GABA in the blood. That was the initial plan. We planned to have MEG and high-density G recordings, also recordings with MRI, transcranial magnetic stimulation was described before as a technique, and the blood draw. The second and the third phase was proved difficult. The kids were difficult to stay still during this recording, so we focused our efforts on the first and the last day of the recordings with the MEG, the high-density G, and the blood draw. That's the experimental setup. Involved like 3 different experiments, the delivery of visual stimuli. More specifically, we presented like flashing checkboards on top of some cartoon figures to make it child-friendly. An auditory experiment where beeps, they were delivered through speakers, and somatosensory experiment where we provide compressed air puffs to the children's fingers. This is an ongoing study, and then this is like the recruitment we have so far. We managed to collect data from 18 children with Dravet and a number of typically developing children or healthy controls. And that's preliminary data from this study that I present today. So this is data from the blood. This is data from 5 kids with Dravet and 5 healthy controls. We did what's called age matching. So we tried to match a child with Dravet with another child who was healthy, a typically developing child with the same age and ideally the same gender. And what we found was quite interesting. In children with Dravet, the GABA level was significantly suppressed. Also, the glutamic acid was also suppressed and several other metabolites. As I said, like this preliminary data, so we are still collecting data. This is like in the same group, we had data from 3 more children with Dravet, but this is like an unbalanced sample in terms of like of the age. The same findings, they remain, significant decrease of the levels of the GABA in the blood of children with Dravet and significant suppression of the glutamic acid. This is the responses to the time frequency analysis plot from the brain. This is like what we collect with high-density EEG. As I explained before, like this is the panels from the healthy controls and the panels for the Dravet kids. At 0 is the timing when we present these flashing checkboards to the kids in front of the screen. The x-axis is where you see the timing of the responses. The y-axis where we see the frequencies in different frequency bands, in alpha, in beta, in gamma frequency bands. And with red color, you see the response to the brain to this kind of stimuli. If you see here, like there's a significant suppression of this gamma activity in kids with Dravet compared to the healthy controls measured by the EEG. We observed the similar finding with the MEG that were performed simultaneously. This is the magnetic activity, suppression of the gamma activity in kids with Dravet compared to the healthy controls. That's findings from the auditory experiment. We provide beeps to the kids with the EEG and the MEG. Although we didn't find statistical significant differences between the 2 groups, you see obviously there's a kind of suppression of the activity in kids with Dravet compared to the healthy controls that was consistent for both the EEG and the MEG. So I would like to close my talk, why these findings are important and what they have to offer us. First of all, they increase the understanding we have about the pathophysiology of kids with Dravet syndrome, as well as other disorders of GABA. Our research also can validate non-invasive systemic biomarkers of brain GABA metabolism. So and that's important for clinical trials in the future. Why we need these kind of biomarkers? Because like when we provide the treatment to the kids through the clinical trials, we don't have to wait for time to see whether the symptoms they improve. We are measuring in the brain what's going on with a non-invasive procedure. So I would like to close my talk by thanking my team. We have a fantastic team at Cook Children's Medical Center. They all work hard on this project. I also would like to thank Dr. Perry for all the support, but most importantly, I would like to thank the families and the parents and the kids who come participate in our studies and supporting our research. It's still an open trial. If you would like to participate, you can find more information to our website at Cook Children's Medical Center. Thank you so much. I know it's a difficult topic, but I will, please.
We have the same question, so
Yes.
I guess I will ask. Thank you for establishing the relationship between the Dravet and GABA suppression. Yeah, so my question as a parent is how can we increase the GABA functionality?
That's a great question. So I don't work with with drugs and drugs development, but I think there's different ways of increasing the GABA. Ways of increasing the GABA through neuromodulation, it was mentioned before as a kind of an approach, is maybe one potential way. Pharmaceutically, I'm not an expert on the field of like how the drugs they act on increasing the GABA level. I think that like there's several companies working on that. There's medications that they act on the GABA receptors, but we don't know why they work sometimes and why they don't work some other times. So there's my way of thinking is like we need better understanding of the underlying pathophysiology. Right now, I think that like we blindly try to understand what's or apply different treatments. It's better like to find out like and have a better understanding what's the underlying pathophysiology in Dravet in order to treat it better.
Right over here. To your right.
Okay,
right here.
good.
It's me.
Sorry. It's really bright here, yeah.
So I may have missed this because I'm trying to monitor like multiple things during your talk, so I apologize if I did, but I guess I'm just trying to, outside of being able to measure responses to medication, do you think this is truly just like a snapshot of in the moment, or do you think that there would be the capability to, the question I get most from parents is prediction of outcomes, and so do you think there's potential for a biomarker such as this in a young child to give any indication of sort of how severe the impairment is and what we might be able to predict long term?
That's a great comment. So I was inspired to start this research when I was back in Boston Children's on the SSAE-DSD. It was an ultra-rare disorder. And I realized that the finding we had on the suppression of the activity was very consistent. But there was also fluctuations among the kids, and we tried to understand why this was happening. So when I moved to Cook Children's Medical Center, and I work with closely with Scott Perry, and we start all this effort with encoded therapeutics and the Dravet, I realized that this is another GABAergic disorder, and then I observed exactly the same phenomenon. So that's an excellent question about whether we are able to correlate the findings we have non-invasive with the MEG or the EEG with the severity of the disorder, and then the prognostic, potentially have a prognostic-like value. We don't know that yet. It takes time for all this kind of research, unfortunately. We do our best like to improve the rates of recruitment and collect the data, but they are quite challenging recordings. And I hope that we'll have more data to analyze. But so far, like what we have, it's quite consistent and seems to be reliable and robust.